Good morning. Thank you for coming. So, as many of you are wondering what is going on with the title of the talk and the presenter this morning, our invited speaker for Stein lecture was stuck last night. I think that there was a pretty bad storm in Dallas, so his flight was diverted to Austin, and then he got stuck in Austin for 8 hours, and then sent me a note last night, well, this morning at 1 o'clock, saying that his flight was actually finally canceled. I don't know why they didn't cancel it earlier, but so he is in Austin at the moment and hoping to fly back to Pittsburgh today. So Dr. Eckel and I were going back and forth last night trying to figure out what to do today because we wanted to make it worthwhile for you coming here. So he graciously agreed to give grand round talk. So we have, instead of a visiting speaker, we have our very own, we are proud of Dr. Eckel. Thank you, Dr. Eckel.
Okay. All right. Hopefully you can hear me well. I'm talking to you a little bit about my research, and that's about circadian rhythms. And hopefully I can show you today how you eventually, by targeting those two circadian rhythms, eventually might be, at least in the near future, could treat some diseases. So first I will talk a little bit about what are circadian rhythms. Then I will show you how circadian rhythms are eventually involved in disease development. And then I will show you what we have learned at the bench and how we think potentially you could treat diseases with it. And to the very end, I will also show a little bit more anesthesia-relevant data from our laboratory, how anesthetics can influence those pathways. So what are circadian rhythms? And that's actually not such an easy question. However, I think there are two important statements that pretty well characterize what circadian rhythms are. So when you think about circadian rhythms, you have to consider that since their existence on Earth, all organisms have been exposed to the cycling of day and night. So that's a very important feature that affects all of us. And because of that cycling, almost all organisms have developed so-called circadian rhythms. And even though this seems to be a very dramatic feature of our lives, the first scientific documentation of circadian rhythms occurred in 1729, and this was done by this French astronomer. His name is Jean-Jacques d'Ortu de Marin, and he was sitting at his desk writing about the stars and got extremely bored, as we sometimes do. And then he looked at his plant and was realizing that the plant and its leaves were moving according to the light conditions. That's the famous mimosa he was looking at. And he all of a sudden had an idea. He thought, what would happen if I would put this plant into a dark room? Would those leaves still move? And he got excited, put this plant in a dark room. And you probably know what happens. Yes, the leaves of this plant were still moving. And so he thought, well, probably he has to repeat that experiment. He bought a new plant and repeated it several times, made sure the room was really dark. And eventually he repeated it again and again. And he finally concluded and said, well, obviously there must be something like an endogenous circadian rhythm in this plant, telling the plant to keep moving. However, he said he has no time at this point to follow up on this. Somebody else will have to do this eventually. And despite the fact that you can see what a dramatic and important feature this is for all our lives, it took another 200 years until researchers again got interested in circadian rhythms. And in this time, people wanted to know if also humans have an endogenous circadian rhythm. This was done by Aschoff. He actually did these studies in Germany and built an underground bunker near Munich. And this is very nicely described in this book by Till Rönneberg, one of his students. He is now a circadian researcher in Munich and very often consulted by hospitals how to build ICUs these days. And Ashoff, so he put humans into an underground bunker, and what they found indeed also humans have an endogenous rhythm. However, it's not a 24-hour time period, it's more a 25-hour. However, he also found that some volunteers showed a 40-hour time period, and this is still not understood until this very day. An interesting and very nice story that's also described in this book. It's a very easy read, by the way, if you're interested. If you think about blind people, some blind people can sense light, so they're completely synchronized to the light environment. However, some blind people cannot see any light. What that means is they live the whole life in a 25-hour day. What that means for them is that every 12 days, their whole their noontime actually shifts to midnight. That means they're wide awake, and that's an explanation why some blind people have severe sleep disorders. And then again, after 12 days, it normally gets better. Today, we believe that the mechanism, how this works, that we actually know the molecular mechanisms pretty well, and this is shown on this slide. I don't want to go too much into detail, but we know that light shines into the eyes and hits so-called melanopsin receptors. The melanopsin receptors are also more recent invention, probably something like 2004, 2006. And when these receptors are hit, they send signals to the suprachiasmatic nucleus and then initiate this complex cascade where clock and B-mail, and don't worry about all these proteins names, they're not that important, but they get increased by light, and then they lead to the induction of other proteins like period and cryptochrome, and they can inhibit their own induction, and eventually this cycle can start all over again. However, I believe today, after all those years, that it is more important to look at this from a different angle to really to understand what circadian rhythms are. I think an important question is to ask the question, what happened 2 billion years ago? And 2 billion years ago, there were two dramatic environmental changes on this Earth. The first thing that happened was that the sun came to Earth. And because of that, there was always, all of a sudden, there was sunlight. And on Earth, there already were those cyanobacteria, these green algae, and they all of a sudden started making oxygen. And oxygen started to accumulate, which eventually led to the Great Oxygen Event. And you can see those were two huge environmental, probably the biggest environmental changes on Earth ever. And based on those changes, and I will show you this in a second, every organism had to adapt to be able to sense light eventually, or to sense oxygen to adapt to those new environmental changes. Interestingly, though, if you think about oxygen, we all appreciate for everyday medicine how important this is for our patients, and if they become hypoxic, we eventually give them oxygen. However, what eventually is less recognized today is that light in this process could be as important as oxygen. And this is, if you want to read it in detail, a review article by Jason. Oh, he sits over there, Jason Brainerd. He did an excellent job on writing this together with Benjamin Scott. This is the same what you saw in a few minutes ago, how light activates those circadian pathways. However, it has this small picture to the side, and what it tells you is that our body has proteins that can sense oxygen, and they're actually related to your circadian or light-sensing proteins. And what I want to tell you with this is that even on a molecular level, those are connected, and potentially, by using light, you eventually could manipulate those pathways, those oxygen-sensing pathways, and eventually use it as a therapy for conditions of low oxygen availability, such as heart attacks, heart ischemia, stroke, sepsis, etc. So in the next few slides, I want to show you how important circadian rhythm proteins actually could be for disease development. Unfortunately, most of the studies have been done in animals, but the reason for that most of the time is that the mouse is the only model that allows us to remove a gene, and with that, you all of a sudden could get a very interesting phenotype. So these, I forgot this. The first I wanted to show you is that circadian rhythms are associated with disease development, has been known since a long time. But this was based mainly on observational or correlational studies, and this might be the most famous study on heart attacks. So it has been known since a long time that heart attacks seem to have a diurnal variation, meaning that you have way more, and might be way more severe heart attacks in the early morning compared to the evening. That's true for many diseases like stroke or stent thrombosis. And I probably could go on and on. There's almost for any disease, a diurnal variation has been described so far. However, if you start looking more on a genetic level, if you knock out those genes, you are all of a sudden surprised that this can lead to real disease development. And this is interesting. This has actually just started basically 10 years ago. So this is pretty new. This is, for example, a Nature paper from 2010. And here they removed one of the clock, or the clock protein from the pancreas in mice and realized that all of a sudden insulin secretion was abolished and they became diabetic. Here, another Nature Medicine paper from the same year showing that another clock protein, the cryptochrome, actually is involved in the production of aldosterone, which eventually can lead to a salt-sensitive hypertension in animals. And here another example, another Nature Medicine, again the same year, cryptochrome also seems to control liver metabolism, in particular gluconeogenesis. If this is not there anymore, you also become diabetic, and eventually explains why, if you have a disrupted circadian rhythm and start eating at night, that this can clearly lead to a metabolic syndrome, as you can see on this picture. Another example, BMAL1, another circadian protein. And here you see what happens if you remove it in mice. They became really big and fat. All right. And then even more, this is more recent research, an immunity paper showing that also inflammation seems to be under the control of the circadian system. And they found that certain anti-inflammatory mediators have a diurnal rhythm. And in fact, if you expose animals to sepsis, they are more likely to die during the late night hours. Another example, BMAL1, if you remove this specifically in the lung epithelium and expose those animals to a bacterial infection, you get a excessive inflammatory response. Nature, again, 2012. Here they showed that the cripple-like factor 15 is under the circadian control, and in fact controls very important potassium channels in the heart that causes, if disrupted, more likely dysrhythmias, in particular at certain time points during the day, and it might be an explanation for the occurrence also of the sudden cardiac death, which also happens obviously more often in the early morning hours. And then another paper, and that was us in 2012, also about period, showing that period eventually might be important during myocardial ischemia, helping the heart to adapt metabolically to the conditions of low oxygenability. We were able to show that certain pathways were able to increase this period 2 during ischemia and help through different mechanisms to perform an oxygen-efficient metabolic pathway. So a quick summary here. So hopefully you can have learned by now how important circadian rhythm proteins can be, that they can influence inflammatory processes, play a role in hypertension, lung disease, obesity, diabetes, or even heart disease. So how do we as researchers come across circadian rhythms, and why did I start studying these proteins. And I have to admit, I had no clue about circadian rhythms 10 years ago. I basically stumbled upon those proteins. I, in fact, was from the adenosine field and started adenosine signaling pathways in the heart. Adenosine is a molecule that has been implicated in cardioprotection since a long time. Already in the early 1900s, people were thinking about adenosine as a cardioprotective molecule. It is, and again, then was reinvented in 1960 by Robert Byrne, a very famous physiologist from Charlottesville, who was again thinking about adenosine in a different context. So if you think about the heart, you have eventually to realize that the heart is a very crazy organ. The heart extracts most of the oxygen already at rest. So that's a problem, because if you exercise, what are you going to do? How do you extract more oxygen? You know, due to the hemoglobin binding curve, that's not possible eventually. So what does the heart do? The heart eventually will start dilating the coronary arteries. In that way, you get more blood delivered to the heart and can eventually have more oxygen while you exercise. And they knew that this was happening. However, nobody knew what causes the vasodilation of the coronary arteries. And his idea, Robert Byrne had the idea, and it was actually an amazing idea. It's unclear how he came about this. He was thinking, well, it must be adenosine, because when you exercise, the first thing you do, you break down ATP, which is converted to ADP and AMP, and then eventually to adenosine. And this was his famous theory, and he eventually was right. And today we know we have characterized all those adenosine receptors. There's the adenosine A1, A2A, A2B, and A3. And just if you were curious, for example, if you push adenosine and cause this heart block when you treat SVTs, that's caused by the A1 adenosine receptor. And his receptor is actually the A2A receptor. The A3 receptor, I don't know if you know that, but if you give adenosine, and if somebody has asthma, you can cause an asthma attack. So this is caused by the A3, and the A2B, in fact, wasn't that well characterized, at least when we started doing our studies in this field. However, more important, based on all these ideas Robert Byrne had, researchers got more interested in nucleotide metabolism or ATP metabolism in the heart during heart ischemia. And this led to something else, another very famous invention in the research world. This was done by Murray in the 80s. He started playing with different ischemia periods and eventually realized if he pre-treated a heart with short, non-lethal ischemic periods, like 5 minutes ischemia, 5 minutes reperfusion, 5 minutes ischemia prior to a longer ischemic period, that he was able to significantly reduce infarct sizes. And actually, he was able to show a reduction by 70%. Probably still until this very day, ischemic preconditioning is probably one of the most powerful cardioprotective mechanism. However, the underlying mechanisms are still not elucidated yet, and hopefully I can show you a little bit what we think how it might work. When I started my research, people knew that adenosine probably plays also a role in ischemic preconditioning. However, the genetic pathways are not well described yet. And so when I met my mentors, the first thing we thought we should do was to establish a mouse model for ischemic preconditioning of the heart. And that's how we do it. You can see we place a small suture around the left coronary artery, and then by hanging weights to the end of the suture and hanging these weights over a rod, you can occlude the coronary artery, as you can see here by the black arrows. And then when you're done, you can slice the heart and stain it. And this is a special staining where the white area indicates infarct and the red area indicates viable tissue. So we used this model, and I brought you a video so you know what we do in the lab. So this is a mouse which is anesthetized with pentobarbital and then intubated with an inside catheter. This is where we prepare the left carotid artery, placing a catheter that allows us to measure blood pressure and to give drugs. And then after the catheter, these are some EKG clamps. They're like, we don't use it anymore. It's not so important to measure, to look at the EKG changes, as we actually can see that we occlude the coronary artery. We also can see when it's reperfused again. This is opening the thorax. It's like a modified technique, so everybody can learn it. It's very hard to see that artery. That's the most challenging part. Unfortunately, those people who made that video were not so good at catching that particular part. Hopefully you can see it a little bit. You see this is an 8-0 suture. It still looks huge, that needle, but it's one of the smallest sutures we have. And then you can see, after we attach some weights to the end of the suture and hanging them over the rod, and then we can occlude the artery. I'm not sure if you can see it. So here's perfused. You see some red flush going through the artery. And then after this is done, you remove the heart and slice and stain it. All right, so after we had established this model, we finally could start working. It took us a while, though. And the first thing we did all of these adenosine pathways. We had mice that were all genetically modified. All these cascades were knocked out. We also then eventually had mice where all the adenosine receptors were knocked out. So we had receptor knockouts for A1, A2A, A2B, and A3. And then we finally realized that the A2B receptor seemed to be the most important adenosine receptor for this cardioprotective effect of ischemic brake conditioning. And then was when I just came here to the United States, 2008. We then also did a screening experiment because we wanted to know what was downstream of this A2B receptor, what was mediating this effect. And as you can see, the top gene that was eventually regulated by our adenosine receptor was period 2, a circadian rhythm protein. And this was in 2008, a little bit irritating, but eventually made sense over the years. And the first thing we did, we believed in our data and started working on mice that do not have period 2. The first thing we did, we looked into heart ischemia, and this is what it looks like if you expose a mouse to 60 minutes of ischemia and 2 hours of reperfusion. You get roughly an infarct size of 45%. However, if you pre-treat that mouse with ischemic brake conditioning, which, by the way, is a longer ischemia time period in a total time, you get this significant reduction in infarct size. However, when we did this in these period 2 knockout mice, we were quite surprised they had really huge infarcts, and on top of that, ischemic brake conditioning did not work. And so early on, the idea was born, how could we target this? How could we influence period? How could we increase period? Obviously, as you have seen in the beginning, those are light-regulated pathways, so the idea was born, well, could we expose animals to light, increase period, and reduce infarct sizes? It sounded crazy, but we did it nevertheless, and we started exposing mice to very intense light, as we realized by searching the literature that the higher the intensity, the more effect you had on the circadian system. And indeed, when we looked, and this is a protein Western blot, don't worry about it, but what it shows you is that over time, if you expose to short, intense light periods, you can see there's an increase of period 2 in the heart. And then finally, we also did ischemia reperfusion studies and found this interesting results. You see over time, we had significant troponin and infarct size reduction in mice after a short exposure to intense light. And if you did the same in the knockout mice, this was completely abolished, telling you that this eventually is a period 2 or circadian specific pathway. The last few years, we spent more time on those pathways and realized that you actually get an even more robust cardioprotective effect if you just keep mice, house them under brighter light conditions. So what we do these days, we have the animals in the vivarium housed under basically daylight conditions, as bright as outside, for 14 hours, and then they will be under dark conditions for 10 hours. So they still have a normal circadian lighting conditions. However, if you do this, you can see over the course of several days, the infarcts become smaller and smaller and smaller. And to give you an idea what the mechanism could be, I brought you some newer data from us. We eventually then, after this intense light exposure, we also looked at period 2 levels in the heart. And as you can see, and this is an interesting phenomenon, and you sometimes read this in the press when they talk about circadian rhythms. So what happens is with intense light, you increase the circadian amplitude. What you get eventually is a more robust and stable circadian rhythm. And if you do this, by the way, in mice that do not have period 2, you cannot increase the circadian amplitude via intense light. So how does it work? After we found this more robust cardioprotection with this intense light housing, we did another screening experiment. We always do screening experiments in research. It is what it is. And so what we found is that eventually the top gene that is regulated by light and also might be period 2 dependent is this so-called angiopoietin-like 4. And this is a paper from Circulation 2012, so obviously it's not an unknown gene. However, it is a gene that has been shown to help to protect the endothelial barrier during heart ischemia. So based on this, we thought probably what we're doing with our light, we're improving the endothelial barrier function. And how can you look at this in our mice? So it's a very simple experiment. What you can do after ischemia, you can just inject Evans blue. Evans blue is very blue, something similar sometimes we use in the OR. Evans blue has binds very strongly to albumin. So if you inject into the vascular system albumin, and then after the experiment is done, you remove the blood, you only should have albumin in the tissue in the case albumin was leaking through the endothelial barrier. You can see if you do 60 minutes of ischemia and then inject Evans blue during the reperfusion phase, you have a significant amount of albumin leaking into the tissue because you obviously significantly cause endothelial damage during ischemia. However, if you do the same experiment to animals that had been under intense light conditions, you see there's a significant reduction of endothelial barrier disruption. And now it looks like a miracle, right, Dan? You're smiling. All right, and this is something we did next. We then eventually thought, well, let's, in a tissue-specific manner, remove period from the endothelium. So these mice have period everywhere, but not in the endothelium. If you do experiments in those mice, and these are the control mice, they are, if you expose them to intense light, this is the lowest light you see, there's a significant infarct size reduction. However, if you do this in mice that do not have period in the endothelium, then there is no protection via this intense light procedure. So this probably indicates that the endothelial period is an important mechanism in this cardioprotective process, and that intense light intense light circadian amplitude enhancement seems to be an endothelial barrier protective effect. Now I'm moving more towards things that might be of interest to you. Over the years, we also thought we should look into how anesthetics could change period 2 expression. This was done actually by Daniel Serd. I'm not sure if he's here. So he actually started those experiments a few years ago, and he exposed animals to all kinds of anesthetics and was curious, since period 2 is such a powerful cardioprotective mechanism, what would happen to those, to period 2 when we do this. And what was pretty striking in those experiments, that always midazolam, and please don't stop using midazolam just based on this data. Okay, this has not been done in humans. This is just in mice, but it's an interesting observation. So you can see midazolam, and this is also seen in different organs, always significantly caused a downregulation of period 2 the cardiac tissue, but also in other tissues. Based on this finding, we thought we should expose mice to midazolam and then look in infarct sizes. And this is where it gets a little bit more dramatic, at least in animals. As you can see, these are troponin levels. They're dramatically increased if you pretreat mice with midazolam, and then after that pretreatment, 2 hours later, expose them to ischemia. So something probably to look into in a clinical setting. We're trying to look into this right now. Is it possible that, and we have something in a clinical setting that's called myocardial injury, would it be possible if patients who have midazolam, are they at higher risk to have this myocardial injury during a non-cardiac surgical procedure? So how could you treat this eventually pharmacologically? A few years ago, the circadian field found these circadian rhythm enhancers. Obviously, they realized how important circadian rhythms are for disease development, in particular for diabetes, for metabolic syndrome. So they started screening all the compounds in the world. And eventually found that this flavonoid, nobiletin, how Nate always says, that this eventually is a very potent circadian rhythm amplitude enhancer and period 2 amplitude enhancer. And so we were curious if by using this nobiletin, if this eventually could undo the effects on the heart we had caused giving mice midazolam. And as you can see, this nobiletin alone significantly increases period 2 levels in the heart and indeed is able to overcome the negative effects of midazolam on period 2 levels in the heart. And if you then do also heart ischemia, you eventually realize that also here you have a phenotype, meaning that if you co-treat mice with midazolam and nobiletin, you again reduce the infarcts. We then also looked at nobiletin alone and saw that this is a very strong cardioprotective agent. If you pretreat animals, you get a significant infarct size reduction, and even might be more interesting, if you do this in our period mice who have not, in our period 2 knockout mice, there is no protection with nobiletin. And we also went a step further and looked at more flavonoid structures to see if this could be something specific, because flavonoids had been described in the past to be cardioprotective. So we looked at all kind of different flavonoids, and interestingly, we only found cardioprotection with nobiletin and tangeritin. And here's the funny part. Tangeritin is another second compound like nobiletin, who was also found to be a circadian rhythm and period 2 enhancer. So, meaning that probably those flavonoids might work through enhancing the circadian rhythm system. And this is the last project I just wanted to show you. Also something that might be more interesting to you. Period 2 eventually could also play a role in the development of delirium. So what we had seen with midazolam in the heart, we also had seen in the brain. And what we found is that midazolam significantly downregulated period 2 levels. And this is a slice of the suprachiasmatic nucleus. You can see there's a significant reduction of period after a single dose of midazolam administration. And since period 2 is an important regulator of sleep, and sleep disruption obviously is something that normally is the beginning of delirium, we thought might be period could play a role in midazolam-induced delirium. And we looked also further in the brain and realized that, in fact, period also is highly expressed in the hippocampus, where midazolam administration also significantly reduced those levels. So how do you study delirium in mice? That's a more complicated question, and we actually had a hard time getting those experiments done and even convince reviewers that this even could be a delirium. Either way, the easiest way to look at a cognitive dysfunction in mice, and we did more experiments. I only want to show you one so you don't get completely exhausted. This is a T-maze experiment. It's very simple. It's so simple that you almost can't believe that it works. So this is a T-maze, and it's a plastic maze. And the animals here, they start at the base, and then they start walking, and the first time they would go left. In the second trial, they would go right. So if a mouse always goes left, something is obviously wrong with that mouse because it cannot remember that it went first left. So this is called spontaneous alternation, and mice normally would alternate at least more than 60 or 80%. And this is what we found after we established this. Saline controls, you see roughly 65% alternation. That's pretty good. You don't get much better probably with animals. However, if you pre-treat mice with a single dose of midazolam and do this 24 hours later, so the mice are not anesthetized anymore, you see we still found a significant cognitive deficit, at least based on those T-maze alternations. And then if you look into mice that do not have period 2, you already realize that they already at baseline with saline have a significantly compromised in their cognitive dysfunction, and however, midazolam had no effect on those animals. So more interesting, and also more interesting for the reviewers, was at some point, can you treat a midazolam-induced delirium? And again, we used this beautiful orange peel-derived flavonoid nobiletin, and first looked if we could undo the transcriptional and changes that we had induced in the brain. And you can see midazolam also downregulates the transcript, not on the protein, and if you co-treat mice with nobiletin, you can completely undo those effects and actually increase period 2 levels. And then we looked at the T-maze, and indeed, we were able to see a therapeutic effects of nobiletin together in conjunction with midazolam compared to the group that had only midazolam. And this was done, by the way, a study with Benjamin Scott, who was a great help here, and a medical student who just left for internship, went to the Mayo Clinic. And I don't want to summarize too much, but I want to show you some of the future and how it could be in the future. I'm not sure if it's necessary in Denver, since we have so many sunny lights, even though I haven't seen them much in the last few days. So this is the Charité, probably one of the biggest hospitals in Europe, and they remodeled their ICU, I think 5-6 years ago, without any reason, just because they thought circadian rhythms are important. And what they built is a very sophisticated system. They have these ceilings that where they can mimic basically dusk and dawn. They can even adapt to the mood of the patient. This is a very complex system, but other ICUs, this is a cardiac ICU in the UK. They also started building these ceilings with daylight lamps. And this is another example that there probably could be something behind this intense light. This is the Karolinska Institute, and they thought they should motivate. Obviously, they have more issues with dark periods, and and so I don't know if you know that, but living in these dark periods eventually causes depression and some sadness. And they wanted clearly to motivate their coworkers and just implemented this intense lightning in all the offices. And indeed, it appears that those coworkers are way happier and more motivated to do their work than without those lightning conditions. And then this is the most important slide for today. These are all the people who worked over the years on these projects. Here, this is one of my postdocs. He's a cardiothoracic anesthesiologist from Japan who's doing actually a postdoc in my lab. And he also just got a American Heart postdoc grant. Would love to recruit him, but of course he's on a J visa and has to leave the country for 2 years. And these are PhD students. She worked a lot on this project, Colleen. Some might know her, whoever attended the AUA meeting a few years ago. She gave a very nice talk. These are graduate students, a lot of PRAs. My mentors, Holger and Sean. This is the division of cardiology, extremely supportive people. You might have seen him recently giving a talk during our research seminar. Michael Bristow, Peter Buttrick, these are from the circadian field. Hoganish, he's sometimes in the press talking about circadian rhythms. He was one of the first cloning all those proteins, BMEL. He's also one of the first people describing those melanopsin receptors in the eye. And then, of course, the most important people, the people from our department. Many thanks to Ben, who always dedicated a lot of time in our manuscripts and helping us troubleshoot and think about the research. Also, Jason Brainerd did a great job writing these review articles on circadian rhythms and hopefully helping kind of educate the anesthesia people a little bit more. And Daniel Surt did an outstanding job on starting those studies on midazolam in the heart, helping me a lot to translate these findings today in a more clinical relevant setting. And then here, important people, Tom Hanthorne, in the past, a lot of excellent support, and today, Paco Hersen and Vesna, who have a really excellent support and very generous support of my research. Thanks a lot. Questions? Now you're tired. Hopefully you can still work in the OR.
Is it only midazolam, or do you look at other benzodiazepines? Characteristic of the benzodiazepine itself, or that particular drug?
Well, you have seen propofol was there as well. We didn't go much deeper. My only explanation would be that midazolam might be the most specific GABA, the most GABA-specific drug we have. And the GABA system is involved in the circadian system. So that has been shown in the past. So it's a very important signaling cascade.
Do you think it's related to the loss of memory? It is also the most potent benzodiazepine we have for recall.
So what I think in terms of the heart, I don't know if you know that, but there are also peripheral GABA receptors. And they're also in the heart. So I believe this is probably a direct effect in the heart causing the downregulation. Then in terms of memory, I'm not sure if I understood that question.
Well, it's just that I'm wondering if some of the similarity, potency for memory and the GABA receptors for that are what is part of the relationship. Because so much of the circadian rhythm is the exposure to light, but for most of us, the exposure to light is through our eyes, so that has a much more potent effect than it does just in our skin. That's what I guess my thought was. There's something about the central nervous system aspects of that.
Could be. I mean, we're still working kind of understand the whole system, how light eventually would induce period in the heart, right? Which seems weird, how light hits the receptors and then how is that signal going? We believe it's through adenosine, and we actually did measurements in tissue. Plasma is always a little bit harder to catch, but adenosine levels go up in the cardiac tissue after these intense light exposures. Yes?
Well, that's a great talk. What's the relationship between sleep and circadian rhythms?
So, as I've said, or briefly mentioned, period 2, if you know that, but that the human mutation of period 2 is also is causing the so-called delayed sleep phase syndrome. That's a syndrome where you actually fall asleep way too late because your melatonin levels do not rise. They normally should rise like 10 p.m., and then you should get tired. Or in those patients, they don't rise until 2 a.m. They fall asleep eventually 3 a.m., then have to wake up at 7, go to work, and then they crash. It's a very disturbing disease. However, it can be treated with intense light, so it's a little bit different. So Does it answer your question, or you wanted to know more?
I guess I was thinking more in terms of the kind of sleep deprivation and the pathway.
Oh, yeah. Yes, I understand what you mean. Yes, if you disrupt the circadian system and you cause sleep deprivation, for example, in humans, you start causing a metabolic syndrome. So there's somebody from Boulder who does all these human studies. He also has, by the way, a big light box in one of the research buildings here. So when you sleep deprive human healthy volunteers, you make them insulin resistant over time. Is that something you, yeah. Yes?
So it's interesting that midazolam is not, this kind of heart condition. And it turned out we have to think about the different way of how we administer anesthetic medications. Because at the same time, we know that midazolam is pro-apoptotic. And in certain conditions, when a patient has a tumor or any nurse has a tumor, midazolam is inhibiting tumor growth. So in a patient who has a cancer, midazolam is actually a very good additional agent. You know what to do today with cancer surgery, how to operate metastasis or reoccurrence. They're just for medications for health. So, for example, midazolam is not used for cardiac surgery in the best cases like 10 to 50 the case. Today, we are not needing so much. It's getting back after this lecture. At least in cardiac surgery, we have the feeling that not tendency is good.
Right. Well, but as I said, I mean, we obviously need to look closer into this before we can make any conclusions. So don't think it's bad drug.
I'm not saying change the
Yeah, right.
I'm just saying we have to change our thinking.
Yeah.
Then we give
Right.
certain medication.
Exactly. And it might also, the time point may play a role when you give it, and all these things are not well understood yet. So obviously there's a lot of room for research, I guess. Good? All right. Thanks a lot.